Good morning dear students. In today's video, let us discuss one of the most important topics, that is Hardy Weinberg's principle, and which is otherwise called concept of genetic equilibrium. And it is the topic from the chapter evolution. And students, if you go through the NCERT textbook, you find that the concept is very simple, because a limited information is given in the textbook, but one of the most important concepts, and we can look into previous NEET papers, definitely there will be a question from this concept, that is concept of genetic equilibrium, given by Hardy and Weinberg. So named after Hardy and Weinberg, it is popularly known as Hardy Weinberg's principle. But if you ask me what exactly this principle is, this principle is otherwise called concept of genetic equilibrium. And coming into the concept, according to Hardy and Weinberg, they explain the concept of genetic equilibrium simply as P plus Q is equals to 1. So according to Hardy and Weinberg, for any given gene in any population, dominant allelic frequency plus recessive allelic frequency is equals to 1. So that's why Hardy-Weinberg principle can be simply written as P plus Q is equals to 1. So with this explanation, it is very clear. In this equation, P represents what? Dominant allelic frequency. Q represents what? Recessive allelic frequency. So P plus Q is equals to 1. First, let us know what is the meaning of this. For any given gene in any population, dominant allelic frequency P plus recessive allelic frequency Q is equals to 1. So let us understand what is this allelic frequency. In simple terms, according to Gregor John Mendel, every gene exists in two alternative forms which are called alleles. So if you ask me what is the definition for allelic frequency, my simple definition is occurrence of a particular allele in the total number of alleles of a gene. And if you imagine 100 genes, there will be 200 alleles, and out of these 200, 100 may be dominant alleles and 100 may be recessive allele. So if I consider dominant allele as capital A, recessive allele as small a, so total number of alleles is 200. In this, dominant alleles are 100 and recessive alleles are 100. So what is the definition for allelic frequency? It is the occurrence of a particular allele in the given total number of alleles of a gene. So in this example, what is the total number of alleles of a gene is given? 200. So capital A is occurring for how many times? 100 times. So small a is occurring for how many times? 100 times. So this 100 times is the frequency of capital A, and similarly 100 times is the frequency of small a. And if I apply it here, so 200 is equals to 1, and ultimately capital A represents what? Dominant allelic frequency. So small a represents what here? Recessive allelic frequency. So if 200 is equals to 1, P will be 0.5 plus Q also 0.5. So this is the meaning of this simple equation, that is P plus Q is equals to 1. And this concept is popularly called concept of genetic equilibrium, given by whom? Hardy and Weinberg. So named after them, it is called Hardy-Weinberg principle. What is this principle says us? For any given gene in any population, dominant allelic frequency P plus recessive allelic frequency Q is equals to 1. Students, till now we understood what is the meaning of P plus Q is equals to 1. And what actually concept of genetic equilibrium in terms of biology. According to Hardy and Weinberg, frequency of a given allele will be under equilibrium for any number of generations. Means according to them, When population size is large, simply we cannot expect the change in the frequency of a given allele of a given gene in a population. So what will be under equilibrium? Allelic frequency of a gene will be under equilibrium when certain conditions exist, they say. So let us understand what are the conditions for genetic equilibrium. So according to Hardy and Weinberg, frequency of a allele of a given gene will be under equilibrium when certain conditions exist. What are the conditions for Hardy Weinberg equilibrium? These are not mentioned in the textbook, so students should listen and try to understand. The first condition, Hardy Weinberg equilibrium is applicable only to a large population. So concept of genetic equilibrium is always applicable to large population. And second condition, population must be random mating population. Random mating population is otherwise called panmictic population. So students should remember this word. Panmictic population means random mating population, means all members of the population should get equal chance to participate in mating, means reproductive isolation should not take place. If reproduction isolation occurs, it is nothing but loss of some alleles and ultimately it results in change in the allelic frequency. So the first condition is population size must be large. And the second condition, population must be panmictic. What is the meaning of panmictic population? Random mating population. And the third condition, all members of the population should be equally fertile. Means, if you have a population of 10,000 rabbits, in that if 100 rabbits are sterile, they never participate in mating. So they are physically existing. Alleles or genes present in them are said to be lost. So because of that, once again, allelic frequency may change. So that's why third condition is that all the members of the population must be equally fertile. And the fourth condition, population should not overlap. So if two adjacent populations overlap, if mating occurs between the members of two adjacent population, it results in gene flow. And once again it results in change in the allelic frequency. So what are the important conditions for equilibrium? Population size must be large and population must be panmictic and all members of the population should be equally fertile and populations must not overlap. When all these four conditions exist, frequency of a allele for a given gene will be under equilibrium, and that was the idea of Hardy and Weinberg, which is popularly known as concept of genetic equilibrium. Now, let us know the factors which can disturb the Hardy Weinberg equilibrium, a very important question. So till now we studied frequency of an allele for a given gene will be under equilibrium when certain conditions exist. But later it was identified that there are various factors which can disturb the Hardy Weinberg equilibrium. So what are the most important factors which can disturb the genetic equilibrium? Here I have written there are 5 factors according to textbook which can disturb the Hardy Weinberg equilibrium. What are the 5 factors? Gene flow, genetic drift, recombination, mutation and natural selection. So these are the 5 factors which can disturb the Hardy Weinberg equilibrium. Means out of these 5 factors, if at least one factor acts on a population, ultimately it results in change in the gene frequency or change in the allelic frequency. So let us study in detail gene flow, which is otherwise called gene migration. The simplest definition for gene flow. What is gene flow? Transfer of genes between the two large sexually reproducing populations. This is the standard definition for gene flow. Let me repeat. What is the definition? Transfer of genes between the two large sexually reproducing populations. Means if you imagine two adjacent populations like A and B. So if members of population A start participating in mating with members of B, so what is happening here? Genes are flowing or transferring from one population to another population, which ultimately disturbs the Hardy Weinberg equilibrium. So gene flow is otherwise called migration, one of the factors which can disturb the Hardy Weinberg equilibrium. And second one, genetic drift, and students should remember this name. Genetic drift is otherwise called Sewall-Wright effect. Means the concept of genetic drift was the idea of Sewall-Wright. So named after Sewall-Wright, genetic drift is also called Sewall-Wright effect. And let me tell you what is genetic drift. So genetic drift is the random change in the gene frequency of a small population purely by chance. This is the standard definition for genetic drift. Let me repeat. What is genetic drift? A random change in the gene frequency of a small population purely by chance, and it was the idea of whom? Sewall-Wright. So named after Sewall-Wright, genetic drift is also called Sewall-Wright effect. So about genetic drift and types of genetic drift, let us discuss in the next video. But now, for time being, let us remember genetic drift is also one more factor which can disturb the concept of genetic equilibrium. And the third one, genetic recombination. You know, crossing over is responsible for genetic recombination. Whenever genetic recombination occurs, ultimately it also results in change in the allelic frequency. And fourth one, mutation. You know already, mutation theory was given by Hugo de Vries. What is mutation? Mutation is the sudden change in the genotype of an organism. So these sudden changes or sudden small changes taking place in the genotype of an organism also can disturb the Hardy Weinberg equilibrium. So mutations are the most important factors which can disturb the Hardy Weinberg equilibrium, because mutations are the sudden changes, they are the random changes. We can never expect in which organism mutation may take place and which gene may be subjected to the mutation. So mutations, according to Charles Darwin, mutations serve as raw materials for variation. So students should remember, mutation is one of the most important factor which can disturb the Hardy Weinberg equilibrium. And finally, natural selection. So whenever natural selection operates on a population, it also results in change in the allelic frequency. Means, according to theory of natural selection, organisms, those who developed positive variations, are said to be fit for survival. Means they have already developed the variation, and what is the source of variation? Mutation is the source of variation. Because of that, whenever natural selection operates on a population, ultimately genetic variation occurs. Reason behind variation is the mutation. Mutation always causes change in the allelic frequency. So with this, I want you to understand the 5 important factors which can disturb the Hardy Weinberg equilibrium are gene flow, which is otherwise called gene migration; genetic drift, which is otherwise called Sewall-Wright effect; genetic recombination, mutation, and natural selection. So we can expect a question like this: which of the following factors does not disturb the Hardy Weinberg equilibrium? Such questions can be framed. So that's why I want you to remember these 5 factors which can disturb the Hardy Weinberg equilibrium. As we all know, on the concept of Hardy Weinberg principle, usually problems are asked in the exams. To solve those problems, first we have to know that we already studied that P plus Q is equals to 1. So what does it mean? For any given gene in any population, dominant allelic frequency P plus recessive allelic frequency Q is equals to 1. When P plus Q is equals to 1, I can write it as P plus Q whole square is equals to 1. So now it is very clear, it is in the form of what? A plus B whole square. So if I write it as No problem, as it is in the form of p plus q whole square, that is a plus b whole square. I have expanded it as what? P square plus 2pq plus q square is equals to 1. So when we write like this, what is the meaning of p square? Means, or what exactly p square represents or 2pq represents in this equation? That is very important. And in this equation, what p square represents? In this expanded form of equation, p square always represents what? Homozygous dominant genotypes of the population or homozygous dominant individuals of the population. Next, 2pq. Pq also represents what? Dominant. But which type of dominant? Heterozygous. So, in the expanded form of equation, p square represents homozygous dominant individuals of the population or homozygous dominant genotypes of the population, and 2pq represents what? Heterozygous dominant individuals or heterozygous dominant genotypes of the population. And ultimately, what is remaining? Q square is remaining. Q square always represent what now? Recessive genotypes in the population or recessive individuals of the population. I need not tell you, recessive phenotypes are always expressed in homozygous conditions. So that's why let us make it very simple and clear. When p plus q is equals to 1, I can write it as p plus q whole square is equals to 1. And expanded form of the equation, p square plus 2pq plus q square is equals to 1. In this equation, p square always represents what? Homozygous dominant individuals or homozygous dominant genotypes of a population. 2pq represents what? Heterozygous dominant or heterozygous dominant genotypes of that population. So q square always represents what? Homozygous recessive genotypes in the population. So if you understand this, we can solve any problem related to concept of genetic equilibrium. Let us solve some 1 or 2 simple problems and understand what exactly Hardy Weinberg principle is. Here is a simple problem I have written. In a population of 1600 rabbits, 256 are recessive. Find out p value and q value. So this problem clearly asking us to find out what? P value and q value. And already we know p represents what? Dominant allelic frequency. And q represents what? Recessive allelic frequency. So for this problem we have to calculate dominant allelic frequency and recessive allelic frequency, that is p value and q value. So how to solve this problem? It is very simple. In the data what is given? Total population size is given. What is the total size of the population? 1600. And we know already, in any given gene for any population, p plus q is equals to 1. So what does it mean? Total population size 1. Here it is represented with how much? 1600. So what we have to calculate? We should calculate p value and q value. So in the data what is given? Total population size is given, and it is also mentioned that 256 are recessive. So recessive represents what here? Q square. Means in the data total population size is given and q square value is given. With the help of this we have to calculate p value and q value. So what is given in the data? It's very simple. Q square is given. Let me write like this. Q square is equals to 256 by 1600. So if I write q square is equals to 256 by 1600, if we calculate this, I get the value 0.16. So what is 0.16? Q square value is 0.16. But what I have to calculate? I have to calculate P and Q. So as I have to get Q value, let me keep it under root. So that's why I'll get now what? Q. So when Q square is 0.16, if I have to convert Q square into Q, if I keep it under root, now I'll get the value 0.4. So Q is 0.4 and now no problem. Ultimately I'll come to know what is the value of P, because already I know for any given gene in any population P plus Q is equals to 1. When Q value is 0.4, ultimately P would be 0.6. See, I have solved this problem. So ultimately what I have calculated? I have calculated P value and Q value, means I have calculated dominant allelic frequency and recessive allelic frequency. Let us solve the same problem in little extended way. See, the problem given here, in a population of 1600 rabbits, 256 are recessive. Find out P value and Q value. It was initial and basic level. And if the same question is like, in a population of 1600 rabbits, 256 are recessive, then what we have to find out? Find out the dominant. Already in the question it is given that recessive are 256. In any population, what else will be remaining? Dominant individuals will be remaining, and dominance can be expressed in homozygous condition and heterozygous condition. So how this problem is now? In a population of 1600 rabbits, 256 are recessive. Find out the number of homozygous dominant and heterozygous dominant individuals of the population. So it is very clear, they are asking you to calculate what now? P square value and 2PQ value, because I already told you, when we write it as P square plus 2PQ plus Q square, P square represents what? Homozygous dominant genotypes, and 2PQ represents what? Heterozygous dominant genotypes. So if you have to calculate homozygous dominant genotypes, you have to calculate P square value. If you want to find out heterozygous dominant individuals, you have to calculate 2PQ value. And I already solved this problem. Now I already know the P value and Q value. Now it is very easy for me. P square, how I have to calculate? Already I know that P value is 0.6. So P square 0.6 into 0.6, I get 0.36. And I already know that total population size is 1600, and you know 1600 represents what here? 1. So 0.36 into 1600. So 0.36 into 1600 means 0.36% of 1600. I get the value 576. So in this population, what is the number of homozygous dominant individuals? 576 are the homozygous dominant individuals. And next what I have to calculate? Heterozygous dominant. Heterozygous dominant is always represented with what? 2PQ. Now already I know the value of P and Q. So let me write 2 into 0.6 into 0.4. So I get 0.48, and already I know total population size is 1600. So if I calculate 0.48% of 1600, I get the value 768. So this 768 is the number of what? Heterozygous dominant individuals of this population of rabbits. 576 are what? Homozygous dominant individuals of the population. And already in the question what is given? Number of homozygous recessive individuals is given, that is 256. So if you want to cross check whether what you have solved is right or wrong, you have to add all these numbers now, that is 256 plus 576 plus 768. If you get 1600, what you solved is absolutely right. So if you understand how did I solve this problem, by finding out p value and q value. So I advise all the students, whenever problem is asked related to Hardy Weinberg equilibrium, first you must calculate dominant allelic frequency p and recessive allelic frequency q values. By finding it out, it is very easy to find out p square, 2pq and q square. So this is how we can apply Hardy Weinberg principle to solve some problems related to gene frequencies and allelic frequencies. So in this video I have explained you what is Hardy Weinberg's principle, what is the concept of genetic equilibrium, conditions for genetic equilibrium, and factors affecting or disturbing the genetic